Hello, anatomy students. I'm recording the Chapter 19 lecture, so even if you're absent, you'll be able to take the notes that you need. So we have 3 objectives this week, all related to genetics and understanding human disorders. So just to start, this should be a review from Biology 1. Males and female humans have 23 pairs of chromosomes, so that's 46. The first 44 of them are autosomes, and 45 and 46 are sex chromosomes, X and Y. And no surprise, males have a Y chromosome and an X chromosome, where females have 2 X chromosomes. So typically we have 46 chromosomes, but sometimes we have a disorder where you end up with too many or too few. And so here's an example of some. We have Down syndrome, Poly-X, Klinefelter, Jacobs, and Turner syndromes, and there are different chromosome numbers. We can figure this out from something called karyotyping. Karyotyping is where we display the chromosomes by size. We're basically making a chart of them, and we have to get the cells, they have to divide, and then we pause them in mitosis, and so in metaphase. So when we do this, we are able to sort them and label them by number. So over here, I want you to take a look on the karyotype on the left at chromosome 21 or pair 21. And on the right-hand side, here's pair 21. So you'll see that the one on the left only has 2 chromosomes there, and the one on the right has 3. This individual would be someone who had Down syndrome. I also want you to look for those sex chromosomes, the X and the Y. So you're going to see right here, this individual has 1 X and 1 Y, so this person would be male. So this problem can occur during meiosis 1 or meiosis 2 when chromosomes don't separate correctly. This is how you end up with too many or too few chromosomes. Down syndrome is also called trisomy 21. It was that chromosome 21 that had too many copies. So here's how it happens. This is meiosis occurring, and in the first step here, these should divide equally between the 2 daughter cells. Unfortunately, this one has all the chromosomes, and this one does not. So all of a sudden, we have 48 over here and 44 on the right, so they're not divided equally. Then when they go through meiosis 2, even though they do divide correctly, each one of these eggs has an extra chromosome that it shouldn't. So that would contribute to one of those disorders. It can also happen during meiosis 2, which means it divided normally in meiosis 1, but during that second step, when it divides, this egg is a typical egg, this egg is a typical egg, this one has 1 too many chromosomes, and this one has 1 too few chromosomes. We also mentioned the X and Y. I don't think it's a surprise to you that male child has an X and a Y, and a female child has an XX chromosome. But there are also potential abnormalities there. And here's a list of all the different possible syndromes that you may see. And we're going to investigate online on Thursday a little bit more detail about this. So this is just basic genetic inheritance from Bio 1. Your genes control your body traits, and the individual genes we call alleles. And so if you think back to Mendelian genetics, if you talk about Gregor Mendel and his pea plants, that would be like the capital T, or lowercase t, those dominant recessive alleles. Okay, so if a trait has homozygous alleles, that means that they are the same, like capital T, or lowercase t. Those are homozygous. If an individual has heterozygous alleles, that would be big T, little t, so the 2 alleles are not identical. And here's how we remember the difference between genotype and phenotype. Genotype is your genes. Those are the letters when you're thinking about that, big T, little t. That's the genotype. Your phenotype is the physical appearance, and I remember that PH, phenotype, physical appearance. It's what it looks like. So big T, little t, even though it has one of each trait, one of each allele, it would express as a tall plant because that's the dominant trait. So here's an example of that, and I'm sure you've seen this from Bio 1. It's earlobes attached and unattached. So up top, we have an example of some different moms and dads. There we go. Mom provides either the dominant or the recessive trait, as does dad. And these are the three possible outcomes. Now, this one has two of the same, which means it's that homozygous dominant. This one would be homozygous recessive. And this one would be heterozygous, which means it has one of each. Because the capital E unattached earlobe is the dominant trait, that's the one that would be expressed. All right, we also have sex-linked inheritance, which means that something is only found on the X chromosome or the Y chromosome. And since a male only receives one X chromosome, it is easier for a disorder to be transmitted in that way if it's found on the X chromosome, because there's not another copy of the X chromosome like there would be in a female. So here's an example, and you probably saw this in Bio 1. It's an example of color blindness, very common. And they're showing you different mothers and fathers. So we have mom here, which has a big B, which is normal color vision, and little b, which is color blindness. And then this father is a typical individual, can see color, just in the same way most individuals can. And so you're ending up with a girl who is normal and not a carrier, a girl who is normal but a carrier, a typical boy, and then you end up with a colorblind boy. That's because mom was a carrier, and because he received the Y chromosome, there's no other X chromosome to sort of override that to be dominant. So this is an example of a sex-linked trait. A Punnett square. Punnett squares are super common, and I'm sure you learned them in Bio 1, but I just want to review really quickly. The individual at the top is one parent, and the individual placed along the side is the other parent. And then what you do is you take the letter from the top and you carry it down into the boxes below. And then you take the letters from the left and you carry them over into the boxes to the right. Okay, genetics gets slightly more complex. We have something called polygenic inheritance, meaning there's more than one gene that controls for a trait, and eye color is one of those things. So there's a huge variety of eye color in the world. So pause the video, see if you can find your eye color, and randomly choose yourself a spouse and see what color eyes your children might end up with. We also have another polygenic trait here in skin color. This is what contributes to the impressive variety that we have on Earth, because there are 3 separate genes you get from your mother and 3 separate alleles you get from your father. And so it's a beautiful wide variety. One of the last examples is multiple allele inheritance, and this is how you inherit your blood type. So I is the letter we use for the trait, and then A is what we use for type A blood, B is what we use for type B blood, and I is kind of like a blank. If you end up with little I, that means you get none of those antigens on the surface of your blood cells, and you end up with type O blood. All right, so we have a handful of practice problems here. Pause and see if you can go through the practice problems, and then in just a moment, I'm going to pop up an answer. Okay, so here's my answer. The Punnett square between a parent with heterozygous unattached earlobes, which is at the top, and homozygous attached is on the left-hand side, and here are your possible 4 children. So here's hemophilia. Because it's a sex-linked trait, it's important to use the X and Y when you do your Punnett square and not just the capital H and lowercase h. This is also in our Amoeba Sisters video. And our last question, the type A blood from Jamie could be IA little I, that would express as A blood, and IA would also express as A blood. So we had to do 2 possible Punnett squares to determine the phenotypes and genotypes for their children. All right, number 4, the outward or physical expression of a person's genes for the trait is phenotype. And then the last one, a couple just had their child diagnosed with sickle cell anemia, which is an autosomal recessive trait. Neither parent has the disease. What are the parents' genotypes? Since this is a recessive disorder,